Welcome back. What we're going to do in this video is we're going to look at gluconeogenesis. So we're going to look at gluconeogenesis. And this is just a fancy way of saying that we're going to basically start with what we ended with. And what we ended with in glycolysis was pyruvate. And ultimately what we're going to do is we're going to take pyruvate. We're going to take pyruvate and convert it into glucose 6-phosphate. We're going to convert it into glucose 6-phosphate. Okay. And the good news about gluconeogenesis that makes it really different than other pathways is you don't really have to learn much that's new. Remember that there were only, out of the 10 enzymes in glycolysis, only 3 of them were irreversible. So basically what you should do is you should go review glycolysis, and just know that the only three irreversible steps were hexokinase, phosphofructokinase 1, and pyruvate kinase. Okay, those were the only irreversible steps, and simultaneously they're also the only allosteric enzymes. But the point is that those are the only enzymes that we have to have different enzymes for. And the reason we have to have different enzymes is because those reactions are irreversible. A lot of it has to do with the fact that the free energy change is just so negative in the glycolysis direction, so we have to use different enzymes. But again, the good news is you only have to do it for 3 enzymes. So the other ones it's the same enzyme name. Now, we're going to start with pyruvate. We're going to start with pyruvate, right? We're going to start with pyruvate, and we're going to have to work backwards to phosphoenol pyruvate. We're going to have to work backwards to phosphoenol pyruvate. So first, let me draw the structure of pyruvate. Let me draw the structure of pyruvate. And one thing about this particular pathway is it has to be done in 2 steps. Now, the first reaction is catalyzed by an enzyme called pyruvate carboxylase. This is pyruvate carboxylase. And I don't know if I mentioned this yet. I don't think it was yet. It's in a later video. But there are 2 types of carboxylases in general. There are carboxylases that use carbon dioxide, and there are carboxylases that use bicarbonate. This particular carboxylase is one that uses bicarbonate. And as a result, usually bicarbonate-dependent carboxylases use biotin. They are biotin-dependent enzymes, and biotin is involved in the mechanism. And at the same time, a lot of these carboxylases tend to hydrolyze ATP. And it's not that any part of the ATP becomes involved in the intermediate or anything, but it's just simply the release of free energy that drives the reaction. And so what we end up getting is oxaloacetate. And if you watch the TCA cycle video, you will see oxaloacetate. And let me actually draw the carboxylate out, and you'll see why I do that in a minute. So here's oxaloacetate. And oxaloacetate, recall, is the terminal intermediate in the TCA cycle. And in this case, the oxaloacetate is going to be consumed by another enzyme. And first, let me draw this right here. Let me draw it like this. Let me do it like this. So GDP, and of course there's going to be an attached phosphate. So this is ultimately GTP, right? But I'm abbreviating the GDP component just for simplicity's sake. And the enzyme that catalyzes this reaction is called phosphoenol pyruvate carboxykinase. So this is not an ATP-dependent mechanism. This is a GTP-dependent mechanism. And I think what you'll find about this mechanism is that it's actually fairly straightforward. It's simple enough to where we can do it. We can do it in a video like this. It's not too complex. But essentially, here's what's going to happen. This lone pair is going to kick in here, right? This lone pair is going to kick in there. And what that essentially does is it forces off carbon dioxide. So initially, we're going to lose carbon dioxide, right? That's part of the mechanism of this enzyme. These electrons have to go somewhere, so they're going to kick in here, right? Now, what that's going to do is that's going to force these electrons out. And what they're going to do is they're going to come out here and hit this phosphorus, right? These electrons kick up. These kick back down. It's just a nucleophilic acyl substitution. And this kicks off GDP. So the second part that's lost is GDP. So the first thing that's lost is carbon dioxide, and then GDP is lost. And of course, it's catalyzed by the enzyme PEP carboxykinase. And so what that generates is phosphoenol pyruvate. So recall the structure of phosphoenol pyruvate. So it looks like this. So here is phosphoenol pyruvate. And recall that it's probably the most unstable compound you'll encounter in Biochem 1 and 2. It's very unstable, very high energy. And if we think about it, like remember from the second glycolysis video, remember when we did pyruvate kinase, we were going from something like this that was extremely high energy and unstable to something that's relatively stable. And so as a result, the free energy change was very, very negative, and that symbolized a very spontaneous reaction, a very thermodynamically favored reaction, right? So if I want to reverse that reaction, right? It would be difficult to do that with the same enzyme, right? Because that enzyme was so much favored in the direction of pyruvate. So what we're going to have to do is we have to use this enzyme. And the way we do that is first we have to burn, and actually we have to burn two nucleoside triphosphates, right? We have to burn an ATP, we have to burn a GTP. And the reason we have to do that is we have to essentially, we're going to something that's significantly higher in energy. And so we have to invest some energy to do that, okay? So PEP to pyruvate, very easy reaction, irreversible. To reverse the reaction, we have to go through these two enzymes. Now, what you're going to find is that once we generate PEP, the rest of the enzymes are just the same, right? Until we get to what? Until we get to fructose 1,6-bisphosphate. And actually, I might also mention this at this point, right? This is the hardest one to know. It's two enzymes. You've never seen them before. But actually, you'll find the other two enzymes that we reverse, their reversed enzymes are actually very, very, very easy to learn compared to this. So what do I have? Well, I have this. I have fructose, right? I have fructose 1,6-bisphosphate, right? So let me go ahead and draw the phosphate. This phosphorus I'm going to abbreviate, right? This one I'm actually going to draw out, and you'll see why in a minute. So here is fructose 1,6-bisphosphate, right? And the enzyme that catalyzes this reaction is called fructose 1,6-bisphosphatase. So this is fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase. And before we go any further, in case you haven't heard it already, a phosphatase is the exact opposite of a kinase. A kinase was essentially a phosphorylase. It transferred a phosphate group onto something. Phosphatases are the opposite. They hydrolyze off phosphate. So essentially what's going to happen is water is going to come in here and it's going to hydrolyze off phosphate. And if we look at the mechanism, it's actually really easy. So here's water. Here's its lone pair, right? Do the nucleophilic acyl substitution, right? Pi bond kicks up, kicks back down, and it kicks off, and actually this is going to abstract a proton, it's going to kick off fructose 6-phosphate. So you're going to end up with fructose 6-phosphate. And remember that fructose 6-phosphate is going to be consumed much in the same way that, well, it's going to be consumed by the same enzyme that produces it in glycolysis, and that was what? Phosphoglucoisomerase, right? Phosphoglucoisomerase. And that's going to produce what? It's going to produce glucose 6-phosphate, right? It's going to produce glucose. It's going to produce glucose 6-phosphate. So let me finish drawing this. But essentially what I want to do right now is I want to look back at what we've done. And I just want to say this, that remember, in the case of gluconeogenesis, and I don't know if I mentioned this already, but phosphofructokinase we're assuming is shut off. So phosphofructokinase was that main allosteric regulatory enzyme of glycolysis. So phosphofructokinase is shut off. So notice, and what would also happen in gluconeogenesis is probably pyruvate kinase would be shut off, and also hexokinase would be shut off most likely. So if you think about it, if pyruvate kinase is shut off, PEP won't be consumed by pyruvate kinase because pyruvate kinase is allosteric and it'd be allosterically inhibited. So PEP would build up, and remember, but these over here are a bunch of equilibrium reactions. And so by Le Chatelier's principle, if I load the body up with PEP, it's going to shift mostly towards this direction, right? By Le Chatelier's principle. Well, I'm going to get to fructose 1,6-bisphosphate, but if PFK is shut off, or excuse me, if PFK is shut off, I'm going to produce a lot of fructose 6-phosphate. Well, if PFK is shut off, fructose 6-phosphate is not going to get consumed by PFK, right? Because PFK is shut off. So I build up lots of fructose 6-phosphate, and it gets consumed by phosphoglucoisomerase to produce glucose 6-phosphate. And glucose 6-phosphate is going to react with an enzyme, and that enzyme is called glucose 6-phosphatase. And again, it's the exact same mechanism as fructose 1,6-bisphosphatase. Water's going to come in and hydrolyze off phosphate. So you would have a water here, right? Again, I'm doing the exact same mechanism. That's why I said these last two are a little bit easier than the first one. So water is going to be a nucleophile. It's going to hit the phosphorus. A nucleophilic acyl substitution is going to occur, and these electrons come out and abstract a proton. So what you're left with is glucose. You're left with glucose, and let me go ahead and draw that. You're left with glucose. So what have we seen in this video? Well, when we undergo gluconeogenesis, we're sort of assuming that those three allosteric enzymes in glycolysis are turned off, or at least they're slowed down to an extent where gluconeogenesis can happen, right? And it all has to do with energy charge. So if you need help with energy charge, watch the video on energy charge, and certainly I have a video on the allosteric regulation of glycolysis. But in terms of gluconeogenesis, we're assuming these three enzymes are shut down. So if I were to build up, like I said, if I were to build up phosphoenolpyruvate, right? If I were to build up PEP, if pyruvate kinase is shut down, then PEP won't be consumed by it. So we won't produce pyruvate. So we're going to be building up PEP, and ultimately it's going to be consumed by, what is it? It's going to be consumed by enolase, right? And in that direction, in the direction of gluconeogenesis, PEP is going to be reacted with enolase, and that's going to form what? 2-phosphoglycerate. And then 2-phosphoglycerate is going to be reacted with phosphoglycerate mutase to form 3-phosphoglycerate, right? And then 3-phosphoglycerate is going to react with phosphoglycerate kinase to form 1,3-bisphosphoglycerate, right? And then 1,3-bisphosphoglycerate is going to react with glyceraldehyde 3-phosphate dehydrogenase to form glyceraldehyde 3-phosphate. And that's going to react with dihydroxyacetone phosphate with aldolase to form fructose 1,6-bisphosphate. And then, of course, that's going to react with fructose 1,6-bisphosphatase to form fructose 6-phosphate. But remember, if PFK is shut off, right? If PFK is shut off, there's no chance of fructose 6-phosphate reacting with it. So I don't have to worry about that reaction happening. So if PFK isn't working, then fructose 6-phosphate builds up. And by Le Chatelier's principle, it forces phosphoglucoisomerase to isomerize fructose 6-phosphate into glucose 6-phosphate, right? And so then glucose 6-phosphate reacts with glucose 6-phosphatase. Water hydrolyzes off the 6-phosphate group, and you get glucose, right? And actually, let's just quickly review pyruvate carboxylase and phosphoenolpyruvate carboxykinase. Remember, if I load the body with pyruvate, it's going to be consumed by pyruvate carboxylase, and it's a biotin bicarbonate dependent enzyme. We hydrolyze an ATP and get oxaloacetate. And this is oxaloacetate. And remember, that's an intermediate in the TCA cycle, but it's also an intermediate in gluconeogenesis. And so these electrons kick in, CO2 is expelled, right? And it ends up, the pi bond to the oxygen extracts the phosphate from GTP. It expels GDP, and we end up with phosphoenol pyruvate. And a lot of what you'll find with gluconeogenesis is really just application of Le Chatelier's principle. And again, with Le Chatelier's principle, we are assuming that those three enzymes are shut down. So if we have high energy charge, right? If we have high energy charge, that's going to be an environment that favors gluconeogenesis. If we have low energy charge, that favors glycolysis. Because if we have low energy charge, we need to be making ATP, we need to be making acetyl-CoA, we need to be making NADH. But if we already have all those things, that's high energy charge, and that will end up, those things will allosterically inhibit those three enzymes of glycolysis. And again, those were hexokinase, PFK1, and pyruvate kinase. And so if they're shut down, gluconeogenesis can run ultimately by Le Chatelier's principle. If I'm loading the body up with PEP, it's going to force all those reactions to go backwards toward fructose 1,6-bisphosphate, and ultimately by those two new enzymes and phosphoglucoisomerase, you're going to end up with glucose. So I hope this video helped, and see you in the next video.